Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Anas bin Sulaiman from class Diagnostic 4A. Today, I'm here to present about the monitoring tissue engineering using magnetic resonance imaging. Magnetic resonance imaging (MRI) is a powerful tool that can be used to monitor tissue engineering at all stages, from in vitro culture to in vivo implantation. MRI also can provide detailed information about the structure, function, and composition of developing tissue, which can help researchers to optimize the tissue engineering process and improve the outcome of clinical trial. In order to better understand the role of MRI in tissue engineering, this review will be described the physical basis for MR contrast in developing tissues. Other than that, to highlight recent MRI studies of tissue engineer construct. And last but not least, to outline new areas for future research and development. The operation principle, we have two principles. Principle of tissue engineering and principle underlying image contrast in MRI. The operation principle states tissue engineering seeks to create biological substitute that restore, replace, or regenerate defective tissues and their function. The Bloch equation in MRI explains the physics of magnetic moments, like those of water protons undergoing precision gyroscopic motion influenced by exponential damping. Here are the flow diagram for bone tissue engineering. After isolation from bone marrow, mesenchymal stem cells (MSCs) are allowed to proliferate in vitro. The cells are then seeded into a biocompatible scaffold along with growth and differentiation factors, for example, beta-glycerophosphate and ascorbic acid. The cells also continue to grow and differentiate in an incubator or bioreactor. Upon maturity, the engineered tissue is transplanted into animal or plant. For the block diagram, alright, for the block diagram, it shows a typical MRI system with the component, pulse sequence, and image display highlighted. When an animal, human, or tissue sample is placed in a magnetic field, the magnetic moment of the protons in the nucleus of the hydrogen atom of the water process at the frequency depend upon the strength of the magnetic field. As a result and finding, it shows an illustration of the anatomical structure of a long and cancellous bone and T1 weighted axial MR image. And then T1 over T2 bar chart and high resolution axial MR image for tissue engineer bone over a 4-week period. As a conclusion, advance in tissue engineering will rely on improvement at all stages of the tissue development process, using the cell source, selecting a biological scaffold, and designing the incubation system. Imaging technique, for example, optical imaging, MRI, are important tools for assessing tissue regeneration at each stage of growth. That's all from me. Thank you.